Hi everyone, my name is Kaori Yamada. Thank you for the opportunity for presenting our project at this meeting. Here, I am showing the therapeutic possibility of targeting VEGFR2 trafficking in the blinding eye disease wet AMD. There is no financial conflict of interest to disclose. Wet AMD is wet age-related macular degeneration. It is a leading cause of vision loss for all the patients. High expression of vascular endothelial growth factor (VEGF) is a hallmark of wet AMD. VEGF induces neovascularization from choroidal vessels. The newly formed vessels are very leaky. The blood cells and plasma induce inflammation and damage to the retina and ultimately cause vision loss. The current therapies targeting VEGF are highly effective for wet AMD patients. However, some patients are still not responding, and repeated treatment decrease the effectiveness of anti-VEGF treatments. Then the alternative strategy is switching the drugs or combination therapy with the other drugs which have different mechanisms of action. This is our working model. To develop the new strategy, first we need to know what's going on in the cells. Endothelial cells lining blood vessels sense the gradient of VEGF, migrate toward VEGF. The fastest cell becomes a tip cell to reach the groove migrating toward VEGF. The followers are stag cells. To sense VEGF from the environment, the receptor VEGFR2 needs to be exposed at the cell surface. However, VEGFR2 is frequently internalized and moving between intracellular membrane compartment and cell surface. We found one of the kinesins, KIF13B, transport VEGFR2 to the cell surface, and it is required for angiogenesis. So, to inhibit massive angiogenesis in the disease condition, current therapies are blocking VEGF. Our strategy is inhibiting VEGFR2 trafficking to the cell surface to prevent the response to too much VEGF. KIF13B is a kinesin family molecular motor in the cells. The cell has cytoskeleton, such as microtubules and actin filaments, to form the shape of the cells. Molecular motors like kinesins, dyneins, and myosins transport molecules on the cytoskeletons. Microtubules are like highways. Actin filaments are like local road. To transport molecules for a long distance, kinesins and dyneins are important. To find the molecular motors for VEGFR2 trafficking to the cell surface, we focused on kinesins. We used VEGFR2 as a bait to find the binding proteins by mass spec analysis. Then we found KIF13B. Here is the data showing the role of KIF13B. We stained VEGFR2 on the cell surface. Before VEGF stimulation, VEGFR2 was localized on the cell surface. After stimulation with VEGF, VEGFR2 was internalized and disappeared from the cell surface and appeared again on the cell surface later. However, when we knock down KIF13B, VEGFR2 could not come back to the cell surface. That means KIF13B is required for VEGFR2 trafficking to the cell surface. Based on the function of KIF13B for VEGFR2 trafficking, we developed a small peptide KAI, kinesin-derived angiogenesis inhibitor. KAI disrupts the interaction between VEGFR2 and KIF13B to inhibit the trafficking of VEGFR2 to the cell surface. KAI selectively inhibits pathological angiogenesis, and it does not affect the survival of normal blood vessels. KAI is a cell-permeable, water-soluble small peptide, so we can use it as an eye drop. First, we tested the binding affinity of KAI to VEGFR2 and other receptor tyrosine kinases by surface plasmon resonance. VEGFR2 showed a high affinity to KAI, and its KD is 6.8 nanomolar. The other receptors had negligible or no binding. Control peptide did not show any interaction with these receptors. Then, to apply KAI for wet AMD model, the first question arises. If we use the KAI eye drop at the top of the eye, does it reach to the area of the disease, which is the back of the eye? To test that, we treated the mouse eyes with KAI eye drop. Then we fixed the eyes at various time points and stained the eyes with Alexa 488 streptavidin, as KAI has a biotin tag. 30 minutes after treatment, we detected biotin KAI in choroidal vessels and the area between RPE and Bruch's membrane. Then, to test the efficacy of KAI on the wet AMD model, we used the laser-induced choroidal neovascularization model. After laser burn on Bruch's membrane, neovascularization from choroidal vessels is induced, which mimics the disease progression of wet AMD. We observed neovascularization by optical coherence tomography and fluorescence angiography. Here is the data showing efficacy of KAI. After laser burn, mice were treated with either control peptide, eye drop daily, or KAI eye drop daily from day 1 to day 14. Mice were monitored by angiography and OCT. On day 14, the eyes were fixed and the neovascularization area was assessed by staining of the choroidal flat mount with ILB4. Compared to the control peptide, KAI treatment significantly reduced the area of neovascularization. Here is the quantified data. KAI treatment significantly reduced the area of neovascularization. The group of KAI 5 microgram day 7 mice were not treated until day 7 to wait for the development of the neovascularization. Then they received treatment from day 7 to day 14. KAI was still effective to reduce neovascularization after the disease progression. It is a more clinically relevant setting. The other groups of mice received intravitreal injection of anti-VEGF antibody or IgG control. Similarly, the other group of anti-VEGF day 7 was untreated until day 7 and received the injection on day 7. Of course, current therapy is also effective to reduce neovascularization, but there was more variation among mice due to the potential risks of intravitreal injection. So this data shows that KAI is as effective as anti-VEGF therapy. Then, to confirm the effect of inhibition of VEGFR2 trafficking by genetic model, we use KIF13B knockout mice. Compared to the wild-type control, KIF13B knockout showed a smaller neovascularization area in the laser-induced neovascularization model. Then, we analyzed sprouting from choroidal vessels ex vivo. We isolated choroidal tissue from wild type and KIF13B knockout mice. Then we embedded the tissue in Matrigel. 7 days after embedding, we measured the sprouting area and showed it in the graph. The sprouting was impaired in KIF13B knockout, which is consistent with the impaired neovascularization in KIF13B knockout in the laser-induced CNV model. In summary, we showed that KAI is specific to VEGFR2 and it has a high affinity to VEGFR2. Topically applied KAI eye drop was successfully delivered to the back of the eye. KAI significantly reduced the disease progression in laser-induced choroidal neovascularization model, which is a well-established wet AMD model. KIF13B knockout showed the important role of KIF13B in disease progression of wet AMD and neovascularization from choroidal vessels. So VEGFR2 trafficking is a promising therapeutic target in wet AMD. I appreciate help from my lab members and collaborators, especially Dr. Malik, Dr. Kazlauskas, Dr. Lee, and Dr. Rosenblatt. This work was supported by NIH funding and UIC funding. Thank you for listening.